Development of innovative geochemical tools for understanding historical boreal fire severity. And I'll skip the abstract. People are welcome to read that on their own. Project members. So this is mostly entirely a lab-based project. And the people that are helping predominantly will be working in the lab. And we have people in Delta Lab at GSC Quebec. And we have people in the organic geochemistry lab in Calgary helping us out. And we have two main collaborators in academia and government at INRS and Government of Alberta. So the project summary and objectives. Higher severity forest fires lead to greater overall emissions of carbon dioxide and have the potential to release old legacy carbon and harmful constituents previously buried in deep layers of soil and permafrost. The ability to evaluate boreal fire severity over the recent geological past would provide a much-needed historical context that is currently lacking from modern estimates of burn depth. So through a series of experiments and application using data lake sediment cores, this study aims to develop a novel geochemical approach to determining boreal fire severity over time to inform national policies that promote adaptation and resilience in boreal communities under a changing climate. So the first map that I'll show is just taken from NRCan's Canadian National Fire Database. The map on the left shows the forest fire coverage perimeters for forest fires over 200 hectares in size from 1980 to 2023. And the graph on the right shows the annual burned area in Canada over roughly the same time period. So this was taken from a paper in Science published recently that used the NRCan data. So they looked at severity, and so they found that around 6% of the modeled areas showed an increase in number of days conductive to high severity burning over this time period. And they found an increase in burn severity in northern Canada compared with other regions in Canada, a larger increase, with this trend being more notable in the years after 2001. And of course, as you can see from this graph, quite notable is that the 2023 anomaly, where there was widespread escalations in burn severity across northern Canada. So, examining historical variability in forest fire severity. Just for some reference, there's some key definitions that are required, and this is based upon Keeley from the International Journal of Wildland Fire. Fire intensity. That refers to the physical combustion process of energy release from organic matter, and that includes temperature, for example, and just the overall energy released from the fire. Fire severity refers in general to the loss of or decomposition of organic matter above or below ground. And of course, as you can probably guess, fire severity is positively correlated with fire intensity. Evaluating fire historical variability in forest fire severity is a challenge. So lake sediment cores can provide reliable temporal perspective going back hundreds to thousands of years. And geochemical and isotopic analysis of fire markers combined with classical approaches such as charcoal counting can yield important insight into burn processes. So one important marker that's can be used to examine historical fire severity are polycyclic aromatic hydrocarbons, or PHs. So PHs are found naturally in fossil fuels and relic organic matter, and they're of course also produced by the incomplete combustion of organic matter, whether that's fossil fuel combustion or from forest fires. And we have there's thousands of different types of PHs. And there's the classical, parent, they're called parent PHs, that the 16 group of parent PHs that the EPA uses to define in a lot of studies. Then you have alkylated PHs. You can see here alk PHs, and those are parent PHs with methyl groups, ethyl methyl groups, so various types of additional alkyl groups. And then you have sulfur-containing PAHs. Well, the term that people use widely is PAC, which is polycyclic aromatic carbon. And then you can have other heteroatom type PHs, nitrogen-containing PACs, and oxygen-containing PACs. So much of the focus of our project will focus on pHs and alkylated pHs, these first two groups right here. So previous work we've carried out at the GSC Quebec over the past dozens of years now, 12 years. We developed methods to analyze pHs and to characterize pHs for their carbon-13 and deuterium ratios. So dual, using both carbon and hydrogen analysis of pHs, called compound-specific isotope analysis, is a very powerful tool in environmental forensics. And as long as you can analyze the carbon-13 and deuterium signature values of your N members, you can apply this in mass balance mixing models to quantify sources in the environment. And so, for example, this graph here shows for results for one compound, for phenanthrene, and you can see there's a wide difference, a distinction between various sources that were applied, for example, in the Athabasca oil sands region of Canada. So how do we measure carbon-13 and deuterium isotopes on pHs? It's determined by gas chromatography interfaced with an isotope ratio mass spectrometer. So your sample is introduced generally as a liquid in some solvent, and from an autosampler, and is injected into a gas chromatograph where your samples are separated based on their physical chemical properties and boiling points by GC, gas chromatograph. And then these peaks are in the case of carbon isotopes, they're combusted at 1050 degrees Celsius, and then the CO2 is ionized, accelerated down a flight tube, and then on a magnet deflected, where you have collectors set up to determine the ratios of mass 44, 45, 46. And it's a similar concept for hydrogen isotopes, but it's pyrolyzed at a higher temperature, and you have different collectors set up to measure the masses for hydrogen gas. But one of the main challenges in doing this and carrying this out in samples is getting good separation. That's very important, because if you don't have good separation by gas chromatography, when you end up pyrolyzing or combusting these compounds, if you have a lot of overlapping with various compounds or interfering contaminants, then this carbon-13 or deuterium signal or hydrogen isotope signal you're going to get will not be reflective of the actual compounds of interest. So this step right here, getting clean chromatograms, is very, very important. And unfortunately, real-world environmental samples are often very dirty and require substantial cleanup efforts prior to CSIA, compound-specific isotope analysis. So here on the left, we have the total ion current chromatogram of a sediment extract that's analyzed by GC mass spec before you analyze it in an isotope ratio mass spec to see what you're looking at. And you can see here that this is what's commonly referred to as an unresolved complex mixture. This is a sediment extract, and it's quite dirty. And you can quantify the pHs by GC mass spec, but this chromatogram right here would not be very useful for compound-specific isotope analysis. You can see the various, here I've plotted 8 different pHs or pH groups. So we developed a method in our lab that was led by a postdoc, Samrat Alam, and using high-performance liquid chromatography, HPLC, we were able to remove much of that baseline and the interfering compounds, so we could actually properly and adequately analyze the carbon isotope ratios and deuterium isotope ratios in a wide variety of pHs. Now, this is a huge step in development from previous work, because and here's the isotope ratio mass spec. You can see it's very similar to the chromatograms shown from the GC mass spec. So this is a huge development in the fact that it allows for a much larger number of 4 to 5 ring pHs that can be analyzed by CSIA. And that's particularly important for this project because these larger pHs are often associated with forest fires. So this can provide much greater insight potentially into forest fire processes That's why this method development is very useful for this project. And this method was published recently in Organic Geochemistry. So one other very novel aspect of this project is combining, the classical CSI analysis using GC-RMS with the future of isotope organic geochemistry using Orbitrap high resolution mass spectrometry. So again, interface with a gas chromatograph. So the difference between this approach for analyzing natural abundance isotopes and GC-RMS is that you don't convert your sample to CO2 or H2. So you can directly, using the high resolution capabilities of this instrument, you can actually analyze different isotopologues and different very small differences in the masses between different isotopologues. So you can simultaneously determine carbon-13 and deuterium and multiple substitutions of heavy isotopes with molecules that is clumping that reflect potentially temperature-dependent chemical reactions and physical processes. So it took a long time to get the software for this instrument, and right now we're still in the very early stages of playing with it and getting some data. But we hope to have some real data on pHs in the relatively near future. So just as an example, back to phenyl-3. So before, we can analyze the bulk carbon-13 or bulk deuterium signature on the whole compound. Here you can potentially, as long as we get the method, tweaked enough, you can analyze, using the various isotopologues down, depending upon, of course, the sample type and ability to measure, the mass, the abundances at such small relative abundances. So you don't expect, for example, there to be a lot of this isotopologue in any sample. Anyway, this is work in progress and it's just started. So we do have a study site now chosen for this project. Our first study site, which is at Lac-Vaudreuil in the Abitibi region, Quebec. And this is basically our fieldwork for this project is piggybacking upon other projects. And in this case, this is work carried out under the framework of Josué Jotzi's, that's that guy right here, his cumulative effects project. And so what I'm showing here is just that we've recently analyzed the lead-210, the americium-241, and cesium-137 in the lake sediment cores that were collected last October. And you can see here on the graph on the right shows the various sediment age models that are determined using lead-210. So these are all naturally occurring radioactive isotopes, or that lead-210 is rather, whereas americium-241 and cesium-137, the spike you see here in 1963 corresponds to their peak in the atmosphere due to the above ground testing of nuclear weapons in the 50s and 60s. So you can use this peak right here to refine your age model. And this particular sediment core, this lake, has an excellent age model, which is what we need actually for the work we're doing if you want to get a high resolution of looking at fire marker distributions and isotopic analysis over time. And we are going to analyze C14 in the deeper part of the core in various plant markers, plant fossils, to get a more accurate age as you go further down in time. And that's it. All I have to report for now, and I'd welcome any questions from anybody. Thank you.